Hey, I think we're live. So we got Lori Hayes and Wooly Ignorant out in the chat already. I didn't have my chat turned on. Are you guys with me?
Messages?
Oh, really?
Yeah, did they make?
I am with you, man.
They're out in the video. Oh,
Did they comment? I don't see any comments so far.
We have David. We have Wooly Ignorant. Dr. Dan popped in for just a second.
Yeah, I sent him a link.
Well, hopefully he'll be able to, but you never know.
Yeah.
Oh, there we go. Hey, Willie. Now, for some reason, I can see comments.
Okay. Well, I think, let me see here. Chromo. Should I do Chromo or YouTube?
Hey, Willie. Hey, David. Lori Hayes.
There's something really important we have to address first before we get into the biology stuff. And I just want to show you what my morning is like with snack.
Oh.
So this is snack. Let's see here. Gotta unmute site.
Watching.
Oh. Wow. This is exactly what I go through every morning at 4 o'clock. That. And the fucking kneading. The slamming. He does that. He hasn't done that yet.
He hasn't discovered that yet, huh?
No, he hasn't.
Oh, the day he does. Uh-oh.
That's it. Anyway, thought I'd share that.
That's funny.
That is just so fucking difficult. Stop it.
Stop it.
Okay, so the first thing I wanted to cover is Dr. Dan was talking to SFT for the first time in, I think, a long time.
Finally.
A long time.
Yeah. And I just wanted to cover this part. Now, he's showing the Ventura paper again. When the fuck is that paper from?
You mean Jesse Ventura?
Yeah, you think so.
That's probably more his speed.
Yeah, that was when Minnesota went Trump, except that Jesse Ventura turned out to get really serious and try to do a really good job once he got elected, but it was a shock when he got elected governor. Everybody's like, what the fuck? He really got elected? Because I think he didn't expect to get elected. He was just kind of running, but he actually tried to do a very good job. And, he's got a lot of critics, but no experience at all, right? Anyway, he was the governor of Minnesota for a while. Let's see. Let me see if I can.
Hey, Dave. Hey, Carol. Hey, everyone.
Okay.
Chat.
Let me play this a little bit now. Donnie gets a little pissed off, I think.
Chromosomes in humans and in chimpanzees. I don't want to spend too much time on this because I do want to argue for the functional nature of the so-called fusion site, which I know you disagree with the most. But I would argue, for example, I'm sure you're familiar with the Ventura paper. I know you are. I've seen you guys critique it before. And so there are aspects of human chromosomes and your grade 8 chromosomes, specifically chimpanzee and gorilla chromosomes, that do have some pretty significant differences. Here in this paper, it talks about the presence of these large blocks of subterminal heterochromatin and this lack of satellite DNA around human chromosomes.
Yeah. So what's heterochromatin specifically?
Repeated. It's repeated DNA basically.
Nope. Not necessarily. It doesn't have to be repeated, but structurally, what's heterochromatin?
Structurally, what is heterochromatin? It's basically
what is heterochromatin?
He doesn't know what heterochromatin
of DNA within the chromosomes that is repeated oftentimes. That's my understanding.
It doesn't have to be repeated though. So the characterization heterochromatin is independent of the sequence. It refers to the physical conformation of the DNA.
Okay.
What does that mean? What is heterochromatin?
Well, I think you're playing word games here because it is. Wait, Dan.
So it's called word games when you press them to actually explain what the fuck they're talking about.
I want you to answer the question: why is this DNA? Well, why is this structure within the DNA? These structures within the DNA.
I'm answering. I'm in the process of answering that question, but I think you're making the argument without knowing what the words mean.
Okay, you know how would you define it if it's not just repeated pieces of DNA basically?
I just want to be clear though.
That's my understanding.
Pause for a second, please.
You're wrong. Okay.
Danikins. Okay, I just want everybody to kind of sit and get the meta view of this. Donnie is a former, or maybe even current, used car salesman. He's read all sorts of articles apparently, and he's written things on virtual paper about the topic. But here you have a working, researching PhD in that very topic, and you have Donnie arguing with him about what things are, what things mean.
Right.
He's
a supposed expert on genetics, and he doesn't know what heterochromatin is.
Soak the arrogance and the Dunning-Kruger, or whatever you want to call it, of that situation in. This is laughable.
Thanks for playing this speed. This is funny.
It's like, teacher, you're wrong. My daddy told me that this is what heterochromatin means. So, teacher, you don't know what you're talking about because my daddy said. And in SFT's case, his daddy is Ken Ham and fucking Kent Hovind.
All the AIG, CMI websites.
Right. Anyway. Oh, let's see what else.
So, heterochromatin refers structurally to DNA that is in a condensed state in the interphase nucleus, right? So do you know what those words mean? In the condensed, like structurally it is supercoiled rather than being in a diffuse state, right? So the nucleosomes are not exposed, right? They're supercoiled so that you don't have like access to enzymes that would do things like express genes. Okay, that's what heterochromatin is.
Well, it's thought to be tandem repeats.
It is largely repeats, but
and here's the thing. Listen, here's the thing. You just want to posture because I did give the correct answer, and then you want to get hyper technical, which I'm fine with. You didn't
give the correct answer.
No, I gave I've seen you all talk about heterochromatin before. I've studied heterochromatin. It's primarily thought to be tandem repeats.
No,
and it's condensed pieces of DNA known as satellite DNA. Okay, fine.
No.
Answer the question.
And he doesn't understand really what satellite DNA is. So the term satellite DNA refers to what used to happen when they would centrifuge chromatin, basically, and they would end up with this outlier area, right? That was primarily a lot of really long repeats, that existed in the centromere and in some subtelomeric regions. And the thing about these repeats is that they're the same DNA over and over again. So what happens is every time your cell divides, it will make copy errors on those things, and it will increase the number of them. And so if you look between humans, you're going to find subtelomeric heterochromatin differences. If you look between all of the apes, basically, and chimpanzees, I think, and a couple other species of ape have a tendency to create more copies of those things. And this is Donnie's whole thing, And also Tompkins' whole thing about why chimps are not 90% identical to humans because of these subtelomeric repeats, right? And so he goes on and on and on about this shit. It's just drives me crazy. Anyway, I thought that was amusing. So
Yeah, that's pretty amusing.
yeah. So where do you want to go
any
with
other clips from that one that you found, or was that it?
That was it for that one.
Yeah.
I have not really listened to it all carefully.
Okay.
For some reason, my head explodes after about 10 minutes of listening to Mr. Preachy.
Yeah, I know what you mean. Yeah.
Yeah.
It's just astonishing because, like, heterochromatin, euchromatin, you learn I learned the difference in like late biology, early college classes. Like granted, I'm still in college right now, so that isn't really saying much, I guess. But like, if you go into it, you learn this stuff, and apparently Donnie hasn't given enough time to. That's just funny.
It's obvious that he hasn't spent time with actual textbooks. He spent time basically quickly reading papers for quotable quotes.
Yeah.
And reading creationist websites. And it's just, yeah, there's euchromatin and heterochromatin. And euchromatin kind of gets looped out and the nucleosomes are exposed. And heterochromatin will be clustered around the edges of the nucleus and other structures that basically keeps a certain amount of genes from being transcribed.
Yep.
And in every cell, you're going to have slightly different structures of this stuff. But, what Donnie had heard is that the repeats tend to be tightly supercoiled because we don't want those damn things translating over.
Right. But heterochromatin, the DNA there isn't necessarily the same thing as those tandem repeats. Like, tandem repeats can be part of the heterochromatin, but
Yep.
Oh, cool. You might be able to join. Thanks, Doctor. Or yeah, if you can just give your maybe what we can do is summarize the arguments he makes here. Maybe Dr. Dan can come in a little bit and give his thoughts on that, and then we can go into more detailed analysis, I guess.
Before we get into ERVs, so ERVs, from what we know about them, they are viruses that entered our genome sometimes as much as 300 million years ago, and anywhere from then up to now, we're like with the koalas, we have evidence that a virus entered their genome about 100 years ago, or within the last 100 years, right? Yep. So they could have entered the genome anywhere. Now, the prediction that an evolutionist would make is that you're going to find a lot of these not only ERVs, but transposons of all kinds. You're going to find a lot of them highly degraded because they don't have a function. They're going to basically mutate and nobody's going to care. But the other prediction about evolution, or now this is really important, is that some of these transposed elements are going to mutate and have some effect on our biology, on evolution, right? The evolutionary path is that they will mutate and some of them are going to be functional. Right. And we would expect like 1% of them to be functional, maybe 1 to 10 at the most, at the very outside.
Well, even for those that are functional, and Dr. Dan drives this point home, that some of them are functional, some of them help with viral mimicry functions, stuff like that. But wouldn't we expect viruses to do that?
Yes,
or things that once were viruses to do that? Like that's what viruses do. They keep out competition from other viruses, and like now they're part of the host genome.
Somewhere way back in the history, early history, prior to mammals, basically, there was one ERV that transposed, and it had a few proteins that have to do with the coat of a protein or coat of a virus.
Right.
And those things actually allowed placental mammals to evolve. and that happened a long time ago. And that's the one that he keeps mentioning is
thanks,
the
Dr. Dan.
placental ERV, right? Which is which keeps the mother from rejecting the placenta, and that's called syncytin. And he mentions that all the time. But I mean, like they picked out like 5 or 10 at the most examples of functional ERVs or functional transposons. He only talks about ERVs, which now the other thing is okay, Andrew, I'll let you handle this part of it. He has Raw Matt and John Maddox have a very weird idea about these transposons jumping and then doing some function.
They're like actually jumping between things.
Dr. Dan is trying to join with Restream.
He's trying to join, but we're getting that blank screen, which is kind of scary, kind of foreboding.
Yeah, that means that we're probably not going to hear Dr. Dan's audio because
damn Restream. Yeah. I don't know. I wonder if he's on Chrome.
Yeah, maybe try a different browser. Okay, all your devices. Anyway.
I hope we can get this to work.
Oh, fuck. Somebody finally noticed that I'm not that dumb. Thank you. Thank you, Randy. God damn. Somebody noticed.
Randy needs to get that super mod status.
Oh, yeah, but he has a qualifier.
A little
trippy. Shit. So, God, we're probably going to miss Dr. Dan because of this.
Damn it.
Okay, so today I've got to reach out to Restream and ask him what the fuck is up.
Yeah, because when it costs us a Dr. Dan, that's a major.
Yeah, that is unforgivable.
That's a major throwback. Yeah.
Maybe you can try another browser or something.
Okay, Athena Midnight too. It cost us Athena Midnight. That was a horrible.
Yeah. Well, thanks for at least trying, Dr. Dan. Yeah, we can see if we can get it to work. Hopefully.
No, we can see you, but you're kind of black and white. Oh, now you're colored.
Okay.
Let's see.
All right, we can hear him too.
Hey,
this is great.
Hey, can you hear me?
Yes. Dr. Dan, how you doing, sir?
Hey, Dr. Dan.
What did you do?
I originally tried it on Firefox because Chrome doesn't play nice sometimes, but then it turned out it actually worked on Chrome. So
Firefox and Restream are arch enemies.
Oh, see, now I know. How's everybody doing? It's been a long time.
It has been.
Yeah, thanks for joining us.
What adventures have you embarked on, Dan, here lately?
Yeah, that was perfect timing. I happened to see you all were going, and I saw the first thing I heard when I popped in was the word heterochromatin. It was like, I know what we're talking about.
Yeah, that part was pretty amusing, though.
Did you notice how irritated Donnie got with you?
Yeah, he didn't like that.
No,
he didn't like that at all.
Yeah, when you push them on something, and that's the case with every one of these creationists. When they throw something out, what you have to do is what they're doing is they're kind of like throwing out decoys. They'll throw out 20 things and we'll try to address all 20 things. If you just address one thing and drive their nose into what they said,
they don't
stick on that.
Like that.
Irritated. Yeah.
Yeah. I also let me just compliment Andrew here because a few minutes ago you brought up the thing about the idea that with viral mimicry that herbs would try to suppress other viruses.
Kind of sloppy language there. Sorry.
No, that was perfect. That was I was doing that for me.
Okay.
That's exactly it. Viruses don't want to compete with other viruses. So if you're gonna infect a host, you're gonna have a mechanism to prevent other viruses from doing that. That's how viruses work. They're gonna be selected to do that. Acting surprised, really that's a function is silly. That's what they do.
Right. I like the way you explained it, particularly the other day. I think it was with your discussion with Rebecca a while back, where you were talking about the ENV gene specifically, or just talking about that specifically. If you have that gene part of the host genome, it's now part of the host. It's going to be selected to benefit the host rather than itself
once you're right, because once you're in the host genome, now your fates are intertwined.
Right.
So
you're not really acting.
You want to protect your host because it's your host, right? You don't want anything to kill your host. Yeah, exactly.
Right.
Now, correct me if I'm wrong.
Yeah, I don't want to be roboting or anything. Okay.
Hang on.
Okay, actively transposing.
Can you still hear me? I can't hear anybody.
Uh-oh.
Oh, you can't hear us? What happened?
Oh.
Can you hear me?
Okay. Now I got you. Sorry. This is me being sloppy over here. I hit a button I didn't mean to hit. My desk is a mess right now, and you're
okay. I have a question. So now I had heard that there are no active ERVs, but I know that some are active in colon cancer. But I've heard that there are no active ERVs currently. They're all ALUs, signs, and lines.
Huh. So I know that they're all suppressed. Like all of our complete ERVs, the ones that have like LTR, GAGPOL, ENV, LTR, like the ones that are all, and there's like only a few dozen of those. So those are all like, we don't want those to do anything, right? Because that's a viral genome essentially. So like we keep those locked down. But in cancers, like you lose gene regulation, right? So you have parts of your genome that are being expressed that shouldn't be. You mess with your methylation patterns. So I would be astonished if like some of that stuff didn't reactivate in certain cancers. But we shouldn't like consider that functional, because like the cancer doesn't help you.
No,
that's bad.
Well, this is the thing I find
point to its activity.
astonishing about this whole argument that ERVs are all functional and beneficial. Like we don't want to be all functional because then they act like viruses.
Yeah, and you can actually like resurrect them, right? And make them reactivate as viruses, and that's bad. Like you could do that experimentally, and that's bad because then you have an active virus, messing around in your genome.
Like they want to look at all the benefits of it and ignore all the detrimental things they can do, even when they're activated.
It turns out that that huge family of 700 zinc finger proteins is one of the things that inactivate these viruses. It gloms onto the DNA and basically keeps them
lock it down. Yeah.
Right.
So I see you're like 40 minutes into this video, which I did not watch because what did I miss? What
we haven't gone over any of it yet. So
I can try and summarize the main or the takeaways, the really bad stuff he said, and then you can give your thoughts on it if you want.
I'd love to hear what he said because I remember from last. It was last year sometime when all this herb stuff started. The thing that he came out with was that the ones that are fixed, the ones that everybody has, were created, and the ones that are not fixed, those are not evidence of endogenization occurring. That's not evidence that occurs widespread. That's actual herbs that do not have a function, but the ones that are fixed, those were created, those have functions. That's what he landed on.
He's still repeating that a little bit, but it's gotten a little worse.
That doesn't make any sense. Because the fixed ones are mostly degraded anyway, because they're the oldest, right? So like the older it is, the more widely shared it is, but also the more degraded. So he's saying like a bunch of solo LTRs are like functional for some reason.
Right.
That doesn't make any sense. What are we doing?
I will also note that before we get into the main arguments he makes, it is funny how in this video he does contradict himself a little bit because he actually mentions all the things that these ERVs, the negative side effects they have when they're fully activated and complete, and then he still says all the beneficial things they do when they're functional. Yeah, it's a little funny to me, but the first big point he makes, which I think was kind of, I think all of them are really a doozy, but the first big point he makes is that, as I understood it, since ERVs are considered a type of LTR retrotransposon or related to that category, he's saying that you can't tell the difference between an external retrovirus infection that makes an ERV, and a retrotransposon that's already there and just inserting itself, mobilizing again the genome, and saying that therefore external ERVs or external ERV infections that result in them, they can't be used as evidence for common descent for some reason.
So,
like he's saying that insertion again and again is part of their function, I should say.
So, like, does he think they're still active though?
Yes,
I
he
guess
thinks
so,
that that's how they work.
But then they can't be fixed in the population because everyone's going to have different insertion sites.
Right.
Does he not realize that? If they're
Well, then
active,
he
then mine
talks
are going to be
about-
different from everyone. Like, they're all going to be unique.
Okay, he talks about these preferred areas of insertion, that there are some areas where they're
Right.
more likely to insert.
Right.
He thinks that solves the problem. He
But
thinks
it's
that's
not
very
to the-
specific.
But it's not to the nucleotide.
Right. It's still
It's,
essentially
it's
random.
regions of thousands or tens of thousands of bases that are hotspot, hundreds of thousands, millions. These are genomic regions out of 3 billion that are
Yeah,
like
hotspots and cold
some
spots.
in-
It's
yeah.
not this specific site.
Right. So like
In
some
his
like
mind-
in certain gene specific regions, some in other specific regions, but it's
Right.
not, like you said, not to the nucleotide
Right.
level.
They'll land, within X number of bases upstream of a promoter, or they'll land in regions that tend to be euchromatin because they're gene dense, right? But they're not going to pick a specific spot next to a specific gene.
And this is why I kind of internally gasped when Donnie said this, because I didn't think he would like make this argument. I thought it would be a little more contrived than that, but he actually said there are site preferences and that solves part of the problem.
That doesn't solve the problem at all.
Right.
It doesn't.
I think he actually thinks that placental ERV. I've heard either John Maddox, Ramat, or SFT, or all three say that they think that what happens is the ERV jumps into that part of the genome and then creates this, placental protection. Right?
I've heard that too, and that's just wrong.
Like,
that's not what it is. It's literally a gene that gets expressed that suppresses inflammation in the uterine lining. Like it's not that the ERV mobilizes. It's that there's a gene that gets expressed that is a gene derived from essentially an anti-inflammatory viral gene, which makes sense if you're a virus to have an immunosuppressive gene. That makes sense, right? So it's not that you're mobilizing the virus. It's the gene that we have co-opted. Right. It's not a complete ERV. It is a gene that we have co-opted. It's not under control of a viral promoter. It's under control of a human promoter. It's not within an ERV. It's a gene acquired via horizontal gene transfer. This is so dumb.
I mean, at that point, can you still even call it an ERV or
well, I don't, because syncytin's not an ERV. It is a viral. It's a retroviral gene acquired via horizontal gene transfer. That's not the same thing, right? It's it's a human gene now. It's not a viral gene that we flip on and off. It's not part of an ERV. It's a human gene. We just got it from a virus. Certainly isn't a virus that mobilizes during embryonic development. That's bananas.
So would you like to go into more depth about how we know these are external ERV insertions rather than just them mobilizing themselves over and over again? Like, because I know retrotransposons do mobilize for a bit after they get in there, but don't they stop at some point?
Yeah, so they get early in embryonic development. You have a lot of retrotransposon transcription, and that leads to some mobilization, right? So they're transcribed, and if the enzymes are translated, I'm not sure how frequently that happens, but they could, do the copy paste thing, right, during embryonic development. Now, the reason we know that's not happening with endogenous retroviruses is that if you look at my genome and your genome and Scott and Speed, you look at everyone's genomes, all of our ERVs are in exactly the same places. Right. Exactly the same places to the nucleotide. If they're mobilizing, that's not going to happen.
Right, because they're still inserting themselves. The same probabilities apply.
Exactly. It's probabilistic.
You're just pushing the issue back even further.
Yeah, exactly. You run up against, and he'll say, hotspots and cold spots. Hotspots are not deterministic. Hotspots, you're not going to land here. You're going to land somewhere in this region. So you survey 2 or 10 or 100 or 1,000,000 people. If they were mobilizing, you would have these things in different places.
Right.
Yeah.
Like, they're not mobilizing. This isn't hard. And that's putting aside the fact that we can watch it happen. There are some unfixed human endogenous retroviruses that only some of the population has, that if you were to fast forward thousands or millions of years, assuming we're still around, either those lineages with those non-fixed ERVs are going to go extinct, or the descendants of all the other lineages are going to go extinct and they're going to become fixed.
I think another thing is what you mentioned about the LTR divergence. Like, we can tell when they've inserted themselves, how long they've been there. So we know that they haven't all been inserted and mobilized over or
at the same time, because they degrade. You lose parts, so you get your LTRs with the genes in between, but the genes tend to get deleted, and you end up with either a pair of LTRs with nothing in between, or just a solo LTR. And the great thing is, you can look at the ERVs that we should, like humans, chimps, gorillas, orangutans, right? You can look at this, and the ones that are more widely shared tend to be more degraded because they're older.
Right. And that just happens to match the nested hierarchy.
It just happens to match the nested hierarchy we get based on sequences. Yep. Just happens to. Coincidentally, I'm sure.
Right. Coincidence. All these things match up. Location, amazing, right? Mutations.
Yeah. So Dan, I just was kind of curious, what kind of argumentation and things do you get in your actual classroom situations? How do they compare to the kind of responses you and attempts at correction that you get from people like Donnie and others in the creationist community?
I will tell you, my students are wonderful. Like, people are always surprised when I say this, but like, and I teach freshman bio. I have 1,000 students sometimes. Not quite 1,000. I'll have like 800, 900 students in the fall, and I'll have like 500 or 600 in the spring. I've got like 500 something students right now. They're awesome. They're really good. They ask really good questions. They're putting stuff. We teach evolution in the fall semester, but then in the spring we do a lot of phylogenetics and biodiversity stuff.
Ooh, cool.
And they're reaching back to last semester and asking questions about, okay, if you have this group with this structure, but then they're related to this group, is that homologous or analogous? And they're putting it all together. They're really good. And like, people do a lot of kids these days about college students, but my experience has been that like they're really good and they're really trying. So awesome. Like, give them more credit. Yeah, they're awesome. Great. I love the questions I get from my students. They ask really good questions. We have tons of fun, and someone will pull out a question. There was one, God, what was it the other day? It was something I'd never thought of before. It was about it was something with viruses and like, is there a virus that somebody asked if there were viruses that infect mitochondria. It's a great question. I don't know. Are there viruses that localize to the mitochondria? Mitochondria derived from prokaryotes. They're viruses of prokaryotes, obviously. Are there essentially bacteriophages of organelle? I don't know. It's a fantastic question. Something I'd literally never thought of in my entire life. First year undergraduate comes up with that. Like, brilliant.
That's cool.
Very cool.
Yeah, I love my students. They're great.
So yeah, those were going back to the SFT thing.
Yeah.
So those were the first two arguments about the site preferences, and of course them mobilizing over and over again, which, as you just explained, is kind of nonsense.
Yeah, you would see that in the DNA. You would see that in DNA sequences. It would be so easy.
Right.
It would be staring us in the face that they were mobilizing. They're not.
The other really bad argument, I think, or at least in my opinion, was more related to the nested hierarchies actions. I think if you know anything about first-rate genetics, it's gonna really, or just evolution in general, it's gonna stick out to you. But just for anyone in the audience, but what he was claiming was that chimps and humans and gorillas and all the other apes, for example, are similar morphologically. Right. So we should expect them to be similar genetically, including the retrotransposons in either case, because of separate and common ancestry, that we should expect that in their model as well.
So common design, basically. It's a common design argument. They were designed the same way, therefore they're going to, you look under the hood, it's going to look the same.
Right, but like, that just struck me as odd, because like similar phenotypes doesn't necessarily mean similar genotypes, right?
Well, that and so that doesn't work, because we know that ERVs are not under purifying selection, right?
Yeah.
So like you can mutate an ERV, there's no selection against
most are selectively neutral.
They're selectively neutral, exactly. So if you have separate ancestry, right? You've got humans, you've got all the other great apes, you've got the other primates, you've got rodents, right? Whatever. And you look at those sequences that are created the same in each one, right? Because they're saying, oh, it's similar morphology, whatever, mammals, right? So they're created the same in each one because functionally they're doing the same thing. And then within each kind, right, which forms a clade, right, that's a each kind is a monophyletic group, right, common ancestor, all the descendants.
Right.
You expect to get a nested hierarchy within that clade, right, because they share common ancestry and whatever mutations happen, and they're selectively neutral. So you're going to get your nested hierarchy in there, right? But then across different kinds, that shouldn't happen because the mutations that occur are probabilistic. So if you take the human lineage as separate from the rest of the great ape lineages, the mutations that happen in the human lineage are going to be different from what happens in the great apes, even if they were created identical, because the mutations, the order, which mutations occur over time is not set in stone. It's not deterministic.
Right.
So this is like, honestly, my favorite single piece of evidence for common ancestry and against separate creation is that we have a very specific testable prediction here. You look at sequences that are not under purifying selection, and ERVs are an example of this. And you say, do we get nested hierarchies within the kinds and find that they're uncorrelated between the kinds? It's just all jumbled up. Or, and that's the prediction for separate ancestry, or do you get a nested hierarchy both within the kinds and across the different kinds? So like, do humans nest with the other great apes? Do all the rodents nest together despite being more than one created kind? Do the humans and the rodents nest together more closely than like with other groups of mammals? Because primates and rodents are actually relatively closely related. And the answer is yes. You see this nested hierarchy across kinds that mimics the hierarchies you get based on sequences, like other sequences,
right? Based on their insertion location and the LTR divergence.
Exactly. So that shouldn't be possible under separate creation. It literally is impossible because the mutations are not deterministic, and that's experimentally. Like, mutations are probabilistic. We've confirmed that experimentally. It's not for debate. So how do you explain that pattern in a young Earth framework? The answer is you can't. It's a direct refutation.
I think the best the way he typically comes at it is he starts from the genetic preloaded diversity, but I think that ignores the fact.
Doesn't matter.
Yeah, and I think I may have mentioned this before in a different comment to you, but we tell, like you said, that there are probabilistic biases to these mutations in terms of transitions versus transversions. And stuff like that. We can tell that they are mutations not just put there or preloaded per se, because the pattern of their occurrence matches what we would expect if they are indeed mutations.
And creationists love to, I was just watching a Ken Ham thing, and like creationists love to do like the observational versus historical science.
Right.
But like mutational biases, we can directly observe those. Like in real time, we know that that's what happens. So you can, exactly what you're saying, you can look at these divergences and you can see how they match with the probabilistic nature of mutations, but you still get a nested hierarchy based on the divergence, which would only be the case if two or more things share common ancestry. And you could build those hierarchies across the so-called created kinds, which means they share common ancestry. Creationists have no explanation for that because you could front load them however you want. You can build them at creation with the nested hierarchy in place. The subsequent mutations are going to muddy that pattern because they're probabilistic.
Right.
So you can't explain the current hierarchies in a special creation separate ancestry model. You just it's to the extent there's one single piece of evidence that directly refutes separate ancestry, like that's it. And ERVs are like poster child for that.
Right, as well as other types of transposons in general, basically.
Yep, that 40 some odd % of our genome that's derived from retrotransposons and stuff. Yeah, exactly. Anything, you could do that with any sequences that are not under purifying selection, which is a lot of the genome.
This is actually I have two off-topic questions, if you don't mind, but semi-related.
Go for it. Yeah, I can stick around for another like 10 minutes, maybe.
Okay.
Is that cool?
Yeah. So in terms of ERVs versus other types of LTR retrotransposons, is there a significant gap percentage-wise, or are most ERVs?
Oh, in terms of like percentage of our genome?
Right.
Oh, it's mostly other retrotransposons. So ERVs are 8%, give or take. And I think retrotransposons, not including ERVs.
Sorry, I meant like LTR retrotransposons specifically.
Oh, I'm not sure that when you cut it. Yeah, when you cut it that fine, I'm not sure.
Okay.
I have it. I might have it in Excel somewhere. Give me a second to ask your other question. Give me a second to look it up.
Sure.
I may have that from when I spoke with Fuzz Rana last year. I may have compiled that, but let me take a look.
At all transposons, you end up with like 48% of the genome is like signs and lines.
Yeah.
It's incredibly high. Oh, let me see here. Where do I have?
My other question was related to, since we talked about it last time about codon bias, I've done some more research on it, and I was wondering, is there a way to tell between when codon bias is occurring because of selective pressures versus when it's occurring due to neutral or mutational biases, like just like the mutational biases we were just talking about?
Oh, my heart. Andrew, you have no idea how happy that question makes me, because that's literally what I did in grad school. Viral codon bias. I literally wrote papers on it's not selection, it's mutation. Like one of my papers, like it's not selection, it's mutation. And here's how you can tell. And so there's a couple ways you can look at it. So one of them is that you can look at, it's a measure. So this thing I kind of came up with, it's kind of a loosey-goosey measure, but it's called codon correlation. And you just plot the codon biases of the host against the codon biases of the virus. And like, so if a specific codon is used a certain percentage of the time, you just plot those two numbers against each other. Like easy. And you see, if it's a strong linear correlation, then that's really strong evidence that the virus is under selection to match the host. If it's uncorrelated, then the virus is not under selection to match the host, and you have to invoke some other explanation. So, for example, that alternative explanation might be a mutational bias. So like in single-stranded DNA viruses, overwhelmingly, the most common form of mutation that occurs is a C to T transition. Cytosine deaminates, it becomes uracil, and then on the next replication, it becomes thymine. So you go C to T is the net effect. And what you find in those viruses is overwhelmingly when you have a codon position that is, you have a wobble position where it doesn't matter what the third base is, overwhelmingly, they end with the codon that ends in T. Like it'll be like 80%, 5%, 7%, 9%, like for the other three. So like that's very clearly, it's a mutational bias that maybe there's selection to match the host codon profile, but that mutation pressure happens so rapidly, and the selection for to match your host is relatively weak. So the mutations just overwhelm it, and you end up
Now,
with.
is that generally the case for all organisms, or is that different for different
It
in
depends.
terms of selection?
So when you have high mutation rates, they tend to drive it. So single-stranded DNA viruses and RNA viruses, it's just mutation rates. The single-stranded
Because they're
DNA have
replicating
this.
so fast.
They're replicating so fast, and they experience mutations really fast.
Okay.
It's the combination of the two. So like the single-stranded DNA viruses have this very specific bias that's clear. The RNA viruses, largely, it seems random. They just mutate a lot. And the codons are just uncorrelated with the base composition in the genome, with the host codon preferences. They're just like doing their own thing. And it's probably just mutations. And the selection, one way or the other, is pretty weak. And that's it. And I actually wrote a paper on that exact thing. Isn't that cool? I love when I get to talk about my own papers.
I guess I'm asking, would that be for like something like vertebrates, invertebrates, would that
So like for our codon bias?
Right.
Yeah, so we have so mammals in particular are kind of weird because we really don't like, and also I agree with Scott. We need to mail Andrew his degree right now. Yes, I agree.
Thank you.
But so mammals are weird because we have this thing, our immune systems recognize it's a C, it's called the CpG dinucleotide C followed by G. We don't like that. When we see CG dinucleotides, we tend to flip out a little bit. And
is that related to CpG conversion bias I've sometimes heard of, or is that a different thing?
I'm not sure. That's a good question. I don't know.
Okay, I just heard that term before, and I'm still looking into it.
Yeah, I don't know. But we don't like CpG for whatever reason. So there are codons. It's the arginine is the one that I know, but there are probably others where within the codon family, it's CG. I think it's CGN, right? So it's CG and then anything. I can look that up real fast. In codon, I think it's arginine. Yeah, it's C. Yeah, so the CG family: CGT, CGC, CGA, CC, CGG. That's the codes for the amino acid arginine, and in our genome, we totally suppress those 4 codons in favor of the other 2 codons for arginine. Arginine's one of the ones that has 6, so it's got 4, and then it's got 2 others that are different. We almost exclusively use the 2, right? So I don't know if there's like a systemic thing for our codons in terms of prefer end in C versus G or whatever, but we suppress the CG pattern. So like arginine is the big one that gets caught in that, where you don't want to use the main 4 because our immune system sees CpG dinucleotides and flips out a little bit.
Okay.
Codons are so cool. I really love and care about codon bias and its evolution. There's approximately 12 people in the world who feel that way, but I'm really into it. It's super cool. Sorry,
for something like Drosophila with a large population size, and this will be the last thing I'll ask, is that more selection driven?
I honestly don't know. I haven't looked at I've only looked at viruses and their hosts, so I don't know about like flies kind of doing their own thing. But I will say that based on what I looked at, selection for codon bias tends to be pretty weak. So like maybe with a large population, it would be selectable over long periods of time. Maybe you would think, right? But I don't know specifically.
But like for vertebrates specifically, like it's generally weak, for example.
I only know of a few exceptions to that, like the CpG thing in mammals. Other than that, my understanding is that it's pretty weak. But again, I haven't looked at every species. I've only looked at a handful. Yeah. Okay. But relatively weak, I think, is the rule of thumb. Yeah, the exceptions are going to be where you have like highly biased genomes. Like some prokaryotic genomes are going to be like 70% AT or 70% GC. And then when you get really extreme biases like that, that's going to affect your codon bias, right? You're just going to tend to end in GC more because you're 70% GC. Like that's just going to happen. But yeah, I can't speak to the specifics of like flies or
it's different for different organisms, basically.
It does differ, yeah.
Okay.
Yeah, there's no like one universal codon bias pattern.
Okay, that makes sense.
So, Dr. Dan, are you familiar with the stuff that Sutherland recently found out about codons and like the evolution of codons?
No. What is this?
Okay, very small RNAs. Okay. If they have a sequence on the 3' end that I think it's UCCUA,
Uh-huh.
I can get you a link to the video that he did. Then the 3 nucleotides prior to that, NNN, match the codon translation. And this will preferentially attach an amino acid to this very small RNA molecule. It's unbelievable. And he hasn't published yet on this, but he did a video.
Wait, so that means that it's not, because we're, the way that we've traditionally thought about that, it's essentially there's no necessary biochemical association between this codon and this amino acid. It's essentially random, right? The code that evolved is essentially just by chance. But now we're saying that may not be the case.
Right. It's awesome. Like biochemically ordained, basically, that
this specific codon must match with this specific amino.
I'll find you the video.
And it's mediated by a small RNA.
Small RNAs, and then there's some other information about how that ends up in the anticodon loop at the bottom of the.
It sounds like the 3 would be the anticodon loop, and the small mRNA would be tRNA.
Except they're too close to the 3' end, and I still don't understand. But there's some further work they're doing about how that ends up creating that particular.
How does it get it? Yeah, because the anticodon, it's not at the other end; it's in the middle. Right.
But it's the evolution of those tRNA.
That is so cool.
It is fucking cool.
So if he hasn't published, how do we know about it?
He did a video where he talks about it with somebody else. We actually did a thing on that with Demeter, I think. So Demeter is working on this, and I'm working with Demeter to use this in our model.
Gotcha.
For creating.
Are you guys talking about stereochemical affinities between codons and amino acids and all that?
Yeah, so something happens to where if you have that, like he hasn't checked on other sequences, but then that sequence is a sequence that is typically in tRNAs today, that 3' sequence, that UCCUA, and then the 3 nucleotides prior seem to have the same coding as modern.
It has to match. Oh, that's so cool. All right. Yeah. So it's coming up on 3:00, so I should get going and do actual responsibilities. But this was awesome timing, and thank you, Speed.
Thanks for joining us, Dr. Dan.
Yeah, it's great to touch base with you, Dr. Dan.
Yeah, it's been too long. I miss all of you.
Yeah, we want you back in the community.
I'm still here. I just the thing is I've done all the creation myths, right? And I've done 20 some odd of them, and I'm running out. It's all the same stuff. Every time ICR AIG writes something, it's something old thing that I've already covered. It's they got to come up with something new
material for us.
Come on, make new bad
from
arguments.
DNA to liver. How do you get from a DNA to a liver? How does that happen?
Yeah. Slowly. Slowly and with great precision.
Well, I'm just talking about in just development of a single individual.
Yeah.
I've looked into that liver thing a bit, and that is a very interesting story. The way the liver is structured, it's pretty easy to see how this damn thing evolved. And kidneys too, another one that's really interesting. You just got to look into it.
Yes, Grignard reagent. We were talking about ERVs.
We were, and it was so bad. I just thank you for filling me in on that, Donnie Stream, because it just sounds bad. It just sounds bad, and I put myself through his whole book on that topic, and I can't take any more of it.
Well, I'm glad I'm not the only one who fought that way.
I could never read that book. It just keeps linking me to death, and I don't know what the hell.
The hyperlinks in a physical book are a problem.
Yeah.
The hyperlinks are a problem.
Because number one, how do you get your mouse pointer to get on it? And then
I use my index finger and I keep clicking on them and nothing happens.
Nothing happens.
Yeah.
Anyway, it's bad. Okay, Dr. Dan, you got.
All right, thanks everybody.
Thank you for being here.
Have a good one, y'all.
Yep.
Well, that was just a nice visit, wasn't it?
That was cool.
Yeah.
Well, we didn't offer him any tea. Damn.
That's a
I
shame.
did offer him that Sutherland video, so I've got to get
Oh,
that.
okay. Okay.
I guess I'll make up for it.
So Andrew, this is
Yeah.
a diagram of our genome basically, and how
Right.
what is what, right? So we got retrotransposon fossils; those are the ERVs, 8%. We got lines 20% and signs 13%. So they occupy
Right.
33% of our genome. Then
Yep.
RNA virus fossils, and we got what else we got here? Introns. Let's see. Introns are like 27%. Other intronic, I'm not sure what that refers to. Protein coding genes, 1%, maybe 2%. In some diagrams show 2%.
Yeah. It's probably
And
going to be
here's-
slightly different for different organisms.
Right. And here's the family. Okay, we got LTRs, long terminal repeats, are ERVs. You
Right.
got the GCs, and you've got the copia genes, and then on the other side, the non-LTRs are line signs and PLEs, and those are those are retrotransposons.
What are the PLEs? I haven't heard that one before.
I forget their God. It
Okay.
stands for some weird pan something PLEs. I forgot what it is. Been a while since I've looked at ERVs myself.
Fair enough.
Transposon. Let's check it out. And I just misspelled transposon. Peewee, is that Peewee? Piggyback like element is a PLE.
I feel like I've heard that before, but only sparingly.
Yeah, BAC. I think that refers to bacteria. I'm not sure. So anyway, but that's a really good diagram every time you're studying ERVs, because you can get so fucking lost trying to figure out which species belongs to which. And the DNA transposons, or the class 2s, are on the right, and they're smaller. So, but I have a whole huge fucking thing here on these guys. Trying to find some other. I have some other statistics. ALU, okay, we got that one. Let me go look down here. Oh, this is a nice one. This shows like the evolution of these things. How many, like some of the ones that have been around for a very, very long time, and
and all mammals.
Yeah, the signs.
Cool.
Kind of shows how they map out. This is a little detail on a line. What else we got here? Got something else. Lads and nads. Let's see, 500,000 copies of line 1 retrotransposed in the human genome. 100,000 truncated, plus 4,000 full-length lines. 98,000 HERVs, which is 5 to 8% of our genome. That's what Donnie wants to talk about, is HERVs, and those, as far as I can tell, are not active in any normal manner, right?
Yeah.
So signs 12%.
Degraded.
Yeah, 1.1 million signs. And if you ever look at NCBI, I was trying to figure out how to turn the track on. You can turn on the signs and lines so you could look at them. And if you scrunch a chromosome up, it's all black because there are so many of them. We're filthy with these things, just filthy. Anyway.
I will say, Gnard reagent, if you want to
Grignard.
go back to the earlier Dr. Ann discussion, I feel like we touched on that a bit. So,
yep.
Maybe.
So, Grignard is my favorite reaction because you put a little bit of magnesium in I forget what the fuck it is, but you scratch the magnesium and all of a sudden it just takes off and it eats the magnesium and your test tube looks like it's going to blow up. And it's just an awesome chain reaction. Very cool.
We like to make things explode, don't we?
We love making things explode. Here at Answers in Atheism, particularly Christians' brains or creationist brains. Anyway, I'm missing something.
Yeah, that was the SFT ERV madness. Hope that was enlightening.
The vice nonsense. So, what's the guy's name? I've got his papers that put forth the Vige nonsense. Basically, they were created like they put them in there so that they would create genetic variation, and we could
Right.
end up the doggy kind branching off into the wolf kind.
Yeah,
and the
because
kind-
Donnie talks about that a little bit in the video too.
Yeah.
Of them creating variation after the flood, supposedly.
Yeah, and when you think about it, how the fuck would that work? I mean, well, obviously transposons have contributed to evolution because some of those
Right,
things
yeah.
end up, functional. But that is not a really good method of creating evolution, right? Like if you were designing something, like if you designed your car so that if you touched a button, it would just randomly turn here or there or accelerate or do some random thing with the hope that it would end up doing the right thing. That's not exactly design.
Right.
I mean, and that's the hypothesis.
Well, another thing similar to that was kind of the way Dr. Dan put it. I think also in the discussion he had with Rebecca, basically, why do you put something there that supposedly is going to mimic and defend against viruses before the fall when viruses don't exist yet?
Right,
What kind of design is that?
It don't exist yet.
This is
Why
creating
not
a
just-
problem that you know doesn't need to be there, and then creating a solution, a supposed solution for that problem that also doesn't need to be there.
Pierre Terborg. I'll type that into the public chat. Pierre, how does he spell it? P-E-R. Terborg is the guy that put that forth. And I have a creationist article on it here up.
I think it was also amusing how in the comments section of SFT's video, Redefine Living, that guy, he commented and said something like, "Oh, or skeptics are still using this
You're roboting.
old war." Oh, I am.
Yeah, just first time. Okay.
Uh-oh. Am I still roboting?
No, you're good. You're good.
Oh.
You're rolling.
Did I watch Sal get when was that, David? Or what stream was that?
Oh, that
I don't
was
know what you're referring to specifically.
Yeah, I was just looking at it a few days ago.
But was that the just one-on-one talk they had the other day?
I think it was, yeah.
Okay. I think I saw part of that.
Yeah. Oh God, let's see now. Okay, John Maddox fortunately has left the planet, so he's
Yeah, he doesn't seem to be doing anything recently.
no, he found out he can't make money on the griff. So he left. So we have Cell and we have SFT and we have Raw Matt and we have Jonathan, and yeah, that's about it. We got to cover those guys. So
Oh, on Atheist Edge. Is that a channel? I haven't heard of that channel before.
that was on Atheist
I'll have
Edge.
to-
What?
Yeah, I get a different stream. I'll have to watch that.
Yeah,
Atheist Edge.
that was a different one. Anyway, what else we got?
Well, I did have some Sal bashing I want to do, but I think I want to save that for a separate stream because it kind of gets into the nitty gritty of it.
Cosmology.
Yeah, cosmology stuff. So
Yeah.
kind of a separate topic from today.
Yeah, we were talking a little bit about that yesterday and the whole early galaxy formation and stuff. I was talking to Dr. Tapioca Weasel, and he was discussing it with me. And that was
yesterday.
Whenever I did the thing with Maya, that was Saturday at noon.
Oh, okay.
Maya Atkinson. So he came on and we were talking a little bit about it. But basically, the idea is, what was the makeup of the very, very early universe, 1 second old? What kinds of clusters were already present? And so it was thought that it was all from quantum fluctuation that the galaxy started to form, right? And we may have to adjust that model and take a look at the possibility that there was more information at the Big Bang than we believe there was. It wasn't just homogeneous. It could have been clustered. And he didn't seem to push. I've had that idea for decades, and he didn't seem to push back on it. So I was kind of happy with that. That may be what we're looking at. We don't know what the makeup was, how whether there were areas that were more condensed than other areas, that sort of thing. So,
hey, David just posted the YouTube link to that, or can you put it in the description so I can check it out later?
Yeah, I will.
Please.
I will.
Also, Captive Desk is apparently having someone from Atheist Edge on Thursday.
Cell Cordova versus Grayson. Oh, this is rich. What? Here we go. Cell.
Ooh, 4 hours.
Oh my goodness. So where does this happen? Oh, nice. Oh, this is great. Okay, I'll put this in the description.
Oh, is this the follow-up debate?
It must be.
Maybe.
Must be. Oh, I'll put that in.
Cool. Thanks, David. I'll have to check this out.
Yep.
We also need to go into Sal's channel and see what kind of comments he makes about his debate. Because remember, there's the nice face of Sal that he puts on publicly. And then when he goes into his channel, he somehow twists things to claim victory or that he made these killer points.
Him and Rebecca were hooting and hollering about how they won that debate because they came prepared. And some people said that Leo and Grayson were not as prepared, with the papers. And it's like, okay, like if Leo and Grayson had prepared the papers that basically are in favor of the Big Bang, the debate would have lasted about 125 years.
And their desks would be just stacked on one side and the other with papers.
Yeah. I think we addressed some of those papers, me, Leo, and Grayson did, at least some of them, after the debate.
The after shows, yeah. Yep. Well, we should do a separate show on that again and try to keep tabs because the creationists are really crowing about this James Webb Telescope findings of the early galaxies. And
yep, we'll have to do a follow-up.
Yep. So Grignard reagent Cell is more dishonest than SFT. Actually, if you really get to know him, you'll find out that he's far more dishonest. He's dishonest.
Both have their pros and cons, I guess.
A very clever way. He has a clever dishonesty that's has to do with conceding, and then completely misrepresenting, and then coming back from another side, and it's just horribly dishonest. So, anyway, I hate when people claim Cell is like this. Oh, he's just this warm and fuzzy guy. If you watch his videos, there's one video where somebody rings the doorbell and he gets up and he struts over and he looks at the guy and then slams the door in his face. And you can just see a different side of Cell's personality that I've seen personally that he's turned upon me. But he has a public face that he has worked on with Sanford. Right. I think they rebuilt his image for his current, YouTube about 5 years ago, because prior to that he was known as Slimy Cell and everybody hated him, and he, and he was really not winning any public awards, and so they decided to redefine his image, and he will crack within 5 minutes. So like he did with Gödel's incompleteness theorem when he was slamming the books. If you just press him a little bit, just fucking press.
Or some of this cosmology stuff for that matter.
Well, the thing is, remember in the actual discussion where it was brought up, he kind of, huffed and puffed a little and then, didn't say much else about it. And then on his own channel, he went on a rage.
Yeah.
That's kind of the pattern with him. He gets thrown for a loop, but then to save face, he goes in a rage on his own.
Yeah, it's his personality. He does that with that church that he's suing or he's involved in. I mean, you can see what he's doing. It's just so goddamn immature. That's what it is. Anyway, okay, you want to wrap her up?
Sure.
Okay.
Unless you got anything else, I don't really.
No, we can always do more ERVs with Donnie. at a later date and come more
Yeah,
prepared.
I think we did a good summary of it today.
Yeah. Yep. As good as we can. Okay,
And
guys.
thanks as always, Dr. Dan, for coming on. Thanks everyone in chat for joining us.
Thank you. And chat. So we had Stephanie Helms, we have Captain Dave from Captain Desk, and we got Grignard Ridge and Carol K. We had Hunter Biden's laptops. Jay was here. Oh, Scott Duke was here. Maya was here. So yeah, we had a few people. So thank you all. Okay, we're gonna end the stream. Ready?
All right.
Goodbye.